Hello, my name is Ryan Lunsford. I oversee particulate testing here at Nelson Laboratories, and today we are providing a seminar on particulates and their role in the pharmaceutical industry. I'd like to thank everybody for being here, and expect this webinar to take about 45 to 60 minutes. To briefly go over our agenda, we would like to start out discussing the concern for particulate matter, why that's a concern to us as manufacturers of pharmaceutical products, also why that's a regulatory concern to the FDA and other regulatory bodies. I'd like to discuss definitions briefly also to go over some of the definitions we might come across when discussing particulate matter testing. Sources of particulates, where particulate matter may originate from or be introduced into a pharmaceutical product. Various test methods for detection and quantification of particulate matter. And then some of the compendial standards that exist out there to help guide us in the pharmaceutical industry, depending on what product we're manufacturing. And lastly, touch briefly on particulate matter identification. Most of this webinar discusses quantification and detection of particulate matter, not so much compositional, but it has become a bit of a hot topic in the last couple of years, so we'll briefly touch on that. First, why the concern for particulates? It may seem elementary, but particulates present in a pharmaceutical product can pose a health risk to patients when exposed to the vascular system. Our vascular system isn't well equipped to deal with large quantities of inorganic matter, especially our kidneys and vasculature are used to dealing with organic bodies, but not so much with foreign material. And so that's the primary concern for particulate matter. The extent of damage or problems that can surface from particulate matter depend on several factors. The size of the particulate and its ability, based on its size, to get caught in different capillary beds or vessels. Also the shape of the particle. It's found that some types of particulate matter, such as fibers, may be of more problematic than, say, round particles. Also the composition, the nature of the particulate and what it's composed of may cause problems with immunological responses or other issues such as an allergic reaction. Also the degree of occlusion. Obviously if a particulate gets stuck in our pinky toe, it may not be as big a deal as if it gets stuck in a capillary bed in the brain. And then the immune response, which we'll discuss briefly in this next slide. Particle implantation in a blood vessel is one of the primary causes for concern about particulate matter, whether that's implantation or actual inclusion or occlusion of a blood vessel. The immunological response can cause a blood clot formation or swelling or other allergic responses or reactions from our immune system. At worst, we could be dealing with an embolic occurrence. At the best, we could be just dealing with implantation, which may lead to plaque formation or other particulate implantation in the future. Also, particulates may become lodged in tissue, which may essentially cause a particulate or foreign matter to gather in our tissues cumulatively and may essentially be there for life. The regulatory concern stems from liability of the aforementioned effects of particulate matter, and also the fact that high levels of particulate matter often indicate insufficient controls or sloppy techniques in your environment or manufacturing process. There are unknown risks with particulate matter. There isn't a great deal of clinical information regarding the effects of particulate matter and foreign material inside the body and in the vasculature. As such, it's often, from a regulatory standpoint, erring on the safe side as a best policy. As manufacturers, obviously we're concerned with complying with regulatory concerns and requirements, but also particulate matter levels of a pharmaceutical product are often an indicator of cleanliness from your environment, where the product may be manufactured, from supplies, whether that be supplies in-house or provided by a third-party manufacturer, manufacturing techniques such as filtration of the product, or any chemistry issues that may exist, such as precipitation of the product under certain circumstances. And handling, packaging, and stability are all concerns if you're seeing a high amount of particulate, that those processes may also be either sloppy or otherwise not sufficient. Typically our PM levels, particulate matter levels, follow other trends in cleanliness and toxicity levels. Here at Nelson Labs, we provide a variety of tests, often back-to-back on the same product. Three of the tests that we frequently perform, in addition to particulate matter, is cytotoxicity, biocompatibility, and TOC, or total organic carbon. Normally, if we see a high level of particulate matter, we may also see high levels of these other compounds that affect these other tests. So particulate matter often tends to go with the same trend as these other tests, which usually are required for a regulatory submission. Now, to go over some of our definitions quickly, particulate matter is defined as mobile, undissolved particles other than gas bubbles unintentionally present in a solution, and that's via the USP 788. We've gone on to discover that from a regulatory standpoint and scientific standpoint, that it's generally accepted that emulsions, colloids, or compounds that are amorphous, such as gels, or dissolve quickly, aren't true particulates. This is important because many of these compounds may cause false positives when detecting particulate matter. We'll discuss this in detail a little further on in the webinar today, but it's important to remember that particulate matter is defined as an undissolvable material. There isn't necessarily a timeline attached to that as to how soon that material would dissolve if given a sufficient amount of time. But typically, if a material can dissolve within a clinically relevant amount of time, it's not considered a true particulate. Now, here's a micrograph of a particle. This is much greater magnification than we typically deal with. But as you can see, they normally exist as somewhat nondescript white or gray chunks. Here's various other micrographs that we've taken here at Nelson Labs. So they vary in size and shape. There aren't any scales associated with these, although we do have some later in the presentation that do have scales associated. Most of these are in the range of 20 to 50 microns in size. As you can see, they differ in color and characteristic. The definition of a fiber, according to the GMP, is a particulate contaminant with a length at least 3 times greater than its width. Fibers are of a special concern because they're much more likely to be caught in the vascular system. They are also likely to gather other materials on them, such as clotting factors or other materials that may cause a blockage or embolistic occurrence, if that's a word, later on. Here's a few examples of fibers. The one on the lower left was stained with Congo red, but as you can see, it's not necessary to stain a fiber in order to see it. They're relatively plain, and most of these are in the range of 100 to 200 microns in length. The visible size range is typically accepted as about 100 microns. Depending on how good your eyes are, it could be as high as 150 or 200, or even down to around 50. Normally, when measuring particulate matter in a pharmaceutical product, we're more concerned with the subvisible range. If we're seeing a lot of visible particulates, we're likely to see a lot more subvisible particulates. So we want to almost eliminate completely any visible particles and focus only on the subvisible. There are a few standards that deal with visible particles, but we're mostly going to be dealing with subvisible at this time, since that's of concern with the USP and the other standards that dictate compliance with particulate matter in pharmaceutical products and injectables. Here's a few micrographs we've taken and went ahead and did some sizing using some software we have in-house. As you can see, particles can range from very small up to quite large. Normally there is a logarithmic increase in particles as you get from large to small. So for every 10 large particles you see, you may see 100 small. And again, it's not really giving a quantitative description, but Typically, as particles get smaller, you see a much greater concentration. That again might be taken for granted, but it's one thing to keep in mind when you're seeing visible particles that there are probably several logs higher of subvisible that you're not seeing. So going on to sources of particulates, there are a great deal of sources of particulates that we have worked with our sponsors to help mitigate over the years through various techniques and investigations. Some of the sources that we have seen as we've done investigations with our sponsors, clients, are first the manufacturing materials that are incoming or produced in-house. Obviously, if you're starting out with a high particulate load in your materials to begin with, whether that be different reagents and solutions, whether it be supplies, those are obviously going to be passed on to the final product. The product design. This typically concerns medical device manufacturers more so than pharmaceutical manufacturers, but it can also have a bearing on a pharmaceutical product if precipitation is likely to occur under certain circumstances. If the design of your product is conducive to harboring particulates or generating particulates, then that's obviously a concern. Residual material after manufacturing and cleaning. This has been a big one with us in the last few years, where we have helped some of our sponsors determine if their cleaning process is sufficient. Again, this applies to medical devices and pharmaceutical products alike, where their cleaning techniques to clean vials or stoppers or seals may be insufficient. The effective packaging can affect particulate levels, especially if the packaging is dirty to begin with. Those particulates can be leached into the solutions and detected when testing occurs. Impact of sterilization hasn't been a big deal. It's of definite concern with regulatory. The FDA likes to see most products sterilized prior to testing. However, we usually don't find sterilization impacts particulate levels very much, although we have seen sometimes where shelf storage may be an issue, which we'll come to in a minute. Impact of shipping and distribution can certainly be a concern. If handling is excessive or the packaging is such that it doesn't provide sufficient padding or protectiveness to the containers or product, then additional particulates may be generated during that process. As I mentioned with sterilization, shelf storage is also a large one. The regulatory like to see aged samples in testing. Shelf storage can sometimes increase, or long periods of storage can increase particulate matter. Not certain why that is, and it's only in some products. It's theorized that it may be due to a breakdown of compounds or possible precipitation. Not usually a big deal, but it's good to keep in mind. Personnel, this is probably one of the largest ones, whether it be clinical or in a manufacturing environment or somewhere along the route from the manufacturing facility to the clinical facility. Personnel are probably the largest providers of particulate matter there is. We certainly find that true in our laboratory. Great measures have to be taken, and aseptic technique needs to be trained to provide sufficient protection from personnel, or from the particulates the personnel may provide and contribute to that product. And then lastly, the manufacturing environment. The environment itself that the product is manufactured in or tested needs to be clean. And we'll get into that a little bit more detail. First, the manufacturing process can contribute significant particulate, and particulates generated during that process should be identified and adequate controls in place. That's straight from the FDA. This again may seem like a no-brainer, but you'd be surprised at how often we see many of our manufacturing processes from our sponsors that are insufficient or not very clean per se. We often visit our clients on site to help them determine maybe where their particulate matter may be coming from, as well as other compounds that affect other tests. And it's important to remember that a great deal of particulate matter may be introduced during the manufacturing process, and that this should be well controlled. In order to control the number of particles found on a finished pharmaceutical product, it's important to minimize the particulates the device or product, pharmaceutical product, is exposed to during the manufacturing process. Again, Relatively elementary, but important to remember. Particles from the environment mostly are from airborne particles. This can include a wide variety of material, usually just basic dust. Most particulate matter, really particulate matter in general, is just a fancy word for garbage. Just residual material that's floating around. It can be anything from skin cells to different compounds, plastics, just about anything floating around in the air. So it's important to remember that we need to control our environment. One of the best ways to do that is through HEPA filtration, whether or not the environment in which the manufacturing, storage, or packaging is taking place in is classified or not. HEPA filtration can significantly reduce the amount of particulate matter in the area. Oh, this is one that my manager threw in here. I like it, so I kept it in there. But just say no to dust bunnies. Keep your areas clean. Usually establishing a cleaning procedure for daily or at least a weekly cleaning of the area is important. Using lint-free materials such as lint-free wipes, clean solutions for keeping the area well in control is important. Particulates from personnel. This is a huge one. That's definitely been a concern for many of our sponsors as well as ourselves. Particulates from personnel can come from many different sources, including cosmetics, dry skin, hair, aerosols, and a variety of others. Sneezing, coughing, fibers, soil from clothes, so on and so forth, et cetera, et cetera. There's several controls that can be taking place here, again, which are very common in a manufacturing environment, but are sometimes overlooked. Appropriate gowning, hand washing procedures, and or the use of gloves. Placing restriction on employees with colds, coughs, or skin conditions. This is one we do here at Nelson Labs when you do have an employee who is in a laboratory environment that may compromise a test. They are put on other tasks and duties that are not as critical to the test. Also, the number of people in the area may also be a consideration. Increasing the number of personnel doesn't just increase the amount of particulate matter that's shed, but also increases the amount of turbulence in the air, those types of things. So keeping that to a minimum is important if possible. Particulate monitoring. I just wanted to touch on this briefly. Many of our sponsors already have some type of monitoring in-house. Depending on the nature of their industry, it may be microbiological monitoring or some type of chemical residual monitoring. Particulate monitoring can be helpful if you are experiencing a high amount of particulate matter in your product, or you suspect particulate may be the cause of other tests that may be failing, or concentrations of different compounds that may be high. Keeping a library of possible contaminants in the laboratory, such as machine lubricants, personnel contaminants, etc., may be helpful. Establishing a program for routine testing and evaluation of controls. Many of our sponsors do this with us or do it in-house to continuously monitor. We may be able to trace that back to a certain material or manufacturing process. Getting into measurement methods, modern particulate testing includes two main methods: an automated method using a piece of equipment that's usually calibrated and doesn't contain any kind of human bias, and then a microscopic method, which is a manual method whereby particles are manually counted and enumerated, or manually counted and sized rather, and data provided that way. Both of these methods are equally important, and we'll get into those a little bit more right now. Nowadays, light obscuration, light blockage principle is the primary mode whereby the automated method is carried out. It may seem rather simple on this diagram. It's an engineering marvel, but really there isn't much to it. There's a microcell or a chamber whereby fluid, whether that be fluid from some type of extraction or fluid from a pharmaceutical product, may be drawn up. As that fluid is drawn up, usually by vacuum, an illuminator shines through the fluid and projects an image on a sensor. That sensor can accurately enumerate and size that particle. There are other methods to generate data regarding particulate matter to gain more knowledge of particulate matter levels in our products. but most of them aren't used as frequently with the USP standards we have today. The light scattering method is somewhat common, but it's more common in small particle measurement, not so much the larger sizes we're testing. And to put that into some quantitative terms, by small I mean smaller than about 1 micron. Normally we're concerned from about 10 to 100 microns in the subvisible range, whereas visible is about 100 and above. So we're concerned with anywhere from maybe 5 or 10 up to about 100. Light scattering is more for the smaller ones, smaller than 1 or 2 microns. So not a huge concern, since many of the compounds in our bloodstream are already 1 or 2 microns. So we're not too concerned about those very small particles. The Coulter counter has also been used for particulate measurement, not as frequently as it used to be. More used for biological material now than particulate measurement, but using an electrostatic potential, it does have the ability to at least count, if not somewhat size, particles. The particle detection principle is relatively simple based on this diagram, but there are issues that can arise based on the light blockage or light obscuration principle. As you can see, as a particle passes through a beam of light, that beam of light can be refracted or diffracted or scattered based on the nature of the particle. Say the particle is translucent or transparent, it can still relatively accurately detect that particle. If it's solid, it may absorb the light or reflect it, which also causes the sensor to trigger that image as a particle. The problem that may occur is that other compounds may also display the same phenomenon, whereby it may refract or reflect the light, but not necessarily be a true particulate. An example would be an air bubble. If an air bubble is drawn up into the sensor, that bubble could essentially cause scattering of the light or some type of refraction. The same as colloids. If some type of oil or silicone gets mixed in with water, it can cause the same phenomenon. So that's where the microscopic method comes in, which we'll discuss in a moment. The microscopic particle measurement is relatively basic, uses light microscopy at 100x per the USP, plus or minus 10x. It uses a calibrated graticule so that we can accurately size each particle, and an external light source, which is important because it helps cast a shadow on material on the filter. Many filters have background noise in order to help the lab technician focus in on the filter, but in order to determine if an observed material is a true particulate, we need to see a shadow. And we've seen this slide before, but as you can see, each one of those particles is casting a shadow. The lower right photo, you can see some background noise there of several little black dots and specks and some texture. But as you can see, none of that is casting a shadow. Only that particle, that large yellow one, is actually casting a shadow. So that's how we accurately detect particles when using the microscopic method. There are several advantages to each method. Light obscuration is very repeatable. One of the great advantages to light obscuration is that it's objective and repeatable. Those are probably two of the big ones. You can analyze a solution, analyze it a week later, and it should have about the same counts. So it varies very little. It's also a calibrated piece of equipment. Quality always likes to see that. Most particle counter sensors are calibrated at least twice a year. It has the ability to analyze very quickly a large quantity of samples. It takes about 1 minute or less to analyze a solution for particulate matter per the USP test. Fluid can be further analyzed since the USP test only draws up 20 mils of solution. It draws up 4 or 5 milliliter aliquots. That can be modified so that the entire solution is consumed, or a significantly higher percentage of the solution or volume of the solution. But per the USP, we only need 20 mils. Also, it can count relatively small particles all the way down to about 2 microns. That's a little bit difficult for the microscopic method. Some of the advantages to the microscopic method, however, is a limited ability to characterize particulates. This may be of concern if you're trying to do an investigation as to where the particles are coming from. If you're able to view those particles, you may have a better idea of where they're originating from. Light obscuration is strictly quantitative. It provides only a count and a size. It does not provide any description of the particulate. Microscopic also is not impacted by various material, which I discussed earlier, such as oils, bubbles, surfactants, and biological material. Several of these may cause an issue with the light obscuration method with false positives. It may detect these materials as particulates when they're not true particulates. Also, the microscopic method is relatively inexpensive to set up. It requires only a microscope, some filters, graticule, and an external light source. Some of the disadvantages, however, really which are opposite from what we just discussed. In light obscuration, there are several materials pre-mentioned before that may cause false positive counts. This may cause you to fail your test and cause quite a bit of grief. There is no option for particle identification, and the initial equipment expense is quite high. Particle counters are normally quite expensive. They have a lot of optics and other very sensitive parts, and so they are not cheap. Also, light obscuration does require a minimum volume, which isn't required by microscopic. The microscopic, however, is somewhat subjective. We currently have 4 analysts here at Nelson Labs who size and count particulates using the microscopic method, and we do experience up to about a 10% difference going from one to another when counting the same sample. While that may be accepted, it's significantly more error than we would experience when using the light obscuration method. The labor expense is quite high. It can take us anywhere from 5 to 20 minutes to count one sample, whereas the particle counter, the automated particle counter, does it in about 1 minute. Typically, per the USP, when you're testing a solution, the entire solution is consumed. You can obviously modify this to only test a portion of the solution, but you are not left with any solution that you can further analyze when testing via the microscopic method. Also, in addition to labor, there's the time required to analyze multiple samples. If you had, say, 30 samples and they're relatively dirty at 20 minutes each, that's going to take you all day. And whereas with light obscuration, it'd be done in an hour. Also, it's very difficult, if not impossible, to examine small particles. There are a few standards that require analysis of particles smaller than 10 microns, and these cannot be done with the microscopic method. So addressing some false positives, this has become a larger concern in the last few years, especially as pharmaceutical products and some therapeutic products have become considerably more advanced and complicated. They contain many of these materials that are likely to cause false positives with the light obscuration counter, yet are increasing in availability and presence in many solutions, injectable products. I mentioned some of these earlier, but proteins, they can tend to aggregate, which may cause positive counts, even though the proteins are meant to be in the solution, and may break down or otherwise unfold in a manner that's not harmful to the patient once injected. Colloids, so any type of micelles or vesicles that may form from different oils or surfactants. Lubricants, which also can fall under colloids, and then surfactants, which again can form micelles, bilayers, and other types of compounds that can cause problems. And also translucent material. Anything that is not completely clear, certainly not opaque, but somewhere in between, can also diffract that light in such a way that we may observe some very strange counts. Some measures that we've used in the past to address some of the false positive counts that we've encountered are, first, simply resorting to the microscopic method. The microscopic method is the USP prescribed method for when the light obscuration method fails. The great thing about this is if a sample were to fail light obscuration but pass the microscopic method, typically it's considered to be within compliance of the USP, assuming that some type of justification can be provided. The second option is to accept the light obscuration data, assuming that it's passing. This may or may not be applicable, but in many cases, a product may pass the USP light obscuration test but be considerably out of trend. In some cases, that may be accepted. In other cases, depending on internal SOPs and out-of-trend investigations, that may or may not be acceptable. But in many cases, the false positives aren't enough to actually cause the sample to fail. The third method, and we've helped many of our sponsors develop a method that reduces or eliminates light obscuration interference. This can be through various techniques such as diluting the product to a point where the materials in the sample are not interfering as much, either because they're not in as close a contact or because a sufficient amount of water or other solvent may essentially modify their ability to cause interference or neutralize them, or by taking certain precautions such as keeping a sample cool or heated to eliminate those false positives. But there are several techniques we've used. Those are just a few. But that's a good way to be able to retain the light obscuration test, yet reduce false positives to a point where they're not causing you to fail. And going over some standards and guidance, luckily in the particulate matter pharmaceutical products issue or subject matter, we have quite a few guidance standards that help us out and a few compendia. The most common is USP 788. This is harmonized with EP 2.9.19 as of 2007, and this by far is our most common and frequently tested standard. This includes pretty much all products that are injectable either parenterally or direct into the vascular system, into subcutaneous tissue. Pretty much all encompasses anything that's shot into you with a needle. So that covers most products. There are a few others. On the bottom right-hand side, we have USP 789. This is specific to particulate matter in ophthalmic solutions. So solutions that are either applied to the eye or sometimes even injected into the eye need to follow this standard. USP 787 is a new standard, a new guidance standard that is currently in development. It deals with particulate matter in protein injections, therapeutic protein injections. I will touch on that a little bit more in the next couple of slides. First, the 788, as we discussed, harmonized about 6 years ago with the EP. It applies to vascular and parenteral injections and infusions, includes intramuscular subcutaneous injections, definitely the most common referred to standard for particulate matter. It contains various specifications and acceptance criteria. There's small volume, which is the most common test. Typically, these are single-dose products with a volume of 100 mils or less. When testing these for particulate matter, it's assumed that the entire injection will be exposed to the patient, and therefore data is produced in counts per container so that we have an idea of the total particulate load from that one dose that will be exposed to the patient. There are two tests per the USP, the 1.B, which is for light obscuration, and the 2.B for the microscopic. The second test is the large volume test, and this is used more frequently for IV products and their accompanying accessories, where it's assumed that maybe the entire solution could be exposed to the patient, but not necessarily. These are for those products that have a nominal volume of more than 100 mils, and data is produced in counts per mil, so it's a concentration-based unit rather than an all-encompassing unit such as counts per container. The purpose of this is to show that the concentration of fluid that's being exposed to the patient is relatively clean. The specs for this are much lower than that of the small volume because if a total amount, say 1,000 mils of solution is exposed to the patient, even with a very small particulate amount per mil, it's still going to be a large amount of particulate after that entire volume is exposed or injected into the patient. So these are typically quite a bit cleaner than our small volume products. There's method 1.A, which is for light obscuration, and 2.A for microscopic. Here's some of the specifications for light obscuration and microscopic testing. For small volume, the first thing you might notice is that microscopic specifications or acceptance criteria are half that of the light obscuration. This is a question we get quite frequently as to why that is. My answer, although the USP doesn't really provide a specific answer, is simply that light obscuration is a little more sensitive. It's either more likely to pick up particles that may not be detected under the microscopic method, or because it may generate false positives that the microscopic wouldn't otherwise generate. Again, that's just my theory, but the USP doesn't give a clear explanation for why this is, but That's the way it is. It also is reflected here in the large volume criteria. They kind of rounded down for the 10 and rounded up for the 25, but essentially it's half for the microscopic as it is for light obscuration. And as you can see, these numbers are quite a bit smaller on the large volume than if we go back to the small volume. You're dealing with, counts in the thousands and the hundreds, but that's per entire container. Whereas when we're talking about per milliliter, we want it to be relatively clean for a large volume injectable such as an IV bag or a saline solution or water for irrigation. Addressing 787 and 789 briefly. 787, again, is specific to protein therapeutics and other similar types of products. This is due to their intrinsic small volumes, often expensive doses, and physical characteristics such as higher viscosity or presence of aggregates, especially common with protein or biologic solutions. Analysis is able to take place with a much smaller volume. I believe via the draft 1 mil will be acceptable rather than 25 mils required by 788. It also contains an informational chapter, which should be quite helpful, 1787, to discuss smaller particles and morphological characterization. So it's very probable that measurement of smaller particles, possibly 2 or 5 micron particles, may be required, as well as possibly some morphological characterization of those particles. But again, this is in development currently, should be released for the next volume compendia update, probably next year, I'm guessing in May. USP 787, or I'm sorry, 789 is specific to ophthalmic solutions. And the acceptance criteria is only provided in counts per mil for some reason, which I've not yet completely figured out, but it is only in a counts per mil unit. This can cause problems when a very small volume is provided, because instead of counts per container, we only have counts per mil we can rely on. Any of those who have tested the 789 may be familiar with this problem. But we've helped many of our sponsors overcome some of the issues with 789 when testing very small volumes, and be happy to help. Also, double the allowed particulate matter is allowed compared to 788, so that's good. I guess things don't have to be quite as clean when they're put in the eye as when they're put in the bloodstream. Also, the inclusion of 50 micron particle sizes are required when using the microscopic method. So, a couple little tidbits, almost trivial there, but important if you'll be testing to that standard. To briefly touch on particulate matter composition and identification, here at Nelson Labs, we frequently test many of our products for particulate matter, or many of our sponsors' products for particulate matter, and receive requests to do some type of compositional work, usually based on FTIR or micro FTIR, if they're able to provide a library reference that we can analyze to and be able to tell them if the products they're seeing, or rather the particles they're seeing in their product, are similar or exact to their reference standards. This is a great way to do some type of investigation to eliminate particulate matter or determine where it's coming from, as well as to provide information to regulatory bodies as to the origin of the material or likelihood of the material being a problem in an injectable product. There are several other ways we can identify particulate matter. Some of these which we have in-house, some of which we don't. There's Raman spectroscopy and SEM, scanning electron microscopy, and then HPLC has limited ability to provide identification of particulate matter, as well as mass spectroscopy. So those are various other methods. Personally, I've only used the FTIR and Raman spectroscopy, but usually they're able to provide sufficient information regarding the composition of particulate matter. But it's important to remember that investigations are definitely good to help reduce the amount of particulate matter in your product and to provide information to regulatory bodies. But at the end of the day, the standard's really concerned with the count and the size of particulates, which sometimes may sound silly, because I certainly would rather have a high concentration of some type of organic material rather than a low concentration of something inorganic such as metal or plastic injected into me. But at the end of the day, it's difficult to break those two down. So right now we deal only with size and count when it comes to the standards. Here at Nelson Labs, we're always happy to consult with you regarding your testing, whether that is testing that you'll be doing regarding particulate matter or otherwise. We've been performing particulate matter testing for about 15 years. We have extensive experience in USP 788, analysis of pharmaceutical products, as well as those other products that I discussed earlier, protein therapeutics, biologic solutions, those with genetic material, a wide variety of other products in order to gain compliance with those, whether it be through one method or another, like obscuration or microscopic. We also have extensive experience in testing medical devices for particulate matter. That is another webinar that we should have available if you're interested, but a large portion of our testing is in regard to vascular medical devices, and we're happy to help with those as well, and the related method validations that are usually required by the FDA. Feel free to contact us if you have any questions. Again, my name is Ryan Lunsford. You can reach me at the number provided there, as well as my email address. Stephen Lamb oversees the microscopic testing in our particulate lab, and his information is provided there as well. Tanya Morris is our supervisor that oversees the pharmaceutical laboratory. She has extensive experience in particulate matter as well. She has served on the AMI committee for development of the technical information report provided in 2010, and is also happy to help. Or feel free to contact our sales department if you'd like to get a quote or have further information regarding our facility or test products. I'd like to thank everyone for joining us today. Feel free to contact us with any questions, and have a good day. Thank you.